All right, great. Well, good morning, everyone. Thank you for joining me today in this 8th webinar in the ASL webinar series. My name is Josephine Lambong. I am the Senior Manager of Science and Industry Affairs at the Alliance for Regenerative Medicine. Today's webinar is on the topic of manufacturing strategy, facility design, and operations, and will be delivered by the lead contributor of Chapter 11 of ASL, who I will introduce in just a few minutes. For those who are not familiar, the Alliance for Regenerative Medicine, or ARM, is the leading international advocacy organization championing the benefits of engineered cell therapies and genetic medicines for patients and healthcare systems. As a community, ARM builds the future of medicine by convening the sector, facilitating influential exchanges on policies and practices, and advancing the narrative with data and analysis. We actively engage key stakeholders to enable the development of advanced therapies and to modernize healthcare systems so that patients benefit from durable and potentially curative treatments. As a global voice of the sector, we are representing more than 475 members globally, including emerging and established biotech companies, academic and medical research institutions, as well as patient organizations. I think everyone here knows that manufacturing for cell and gene therapy is a complex process, and it is often the rate-limiting step in the commercialization of regenerative medicines. So the industry right now is in the process of rapidly scaling up, and there are more phase 3 studies and more commercial product approvals anticipated. However, products are being held up at the BLA stage due to CMC issues. So we see that the advances are being made in the manufacturing technologies, and overall the experience in the workforce are improving, which expands the process understanding. However, standards and best practices in the field are not advancing as quickly, and this is the pre-competitive area where ARM focuses our efforts and to further advance the process understanding in the industry to facilitate product development and commercialization. So ACEL is a project that ARM undertook several years ago after feedback from our members about the CMC issues that the field as a whole is experiencing. It is a best practices document which incorporates collaborative case studies presenting the implementation of quality by design principles in cell-based therapies. And to make ACEL an effective resource, we chose to focus on immune cell therapy as a case study, and in particular, an autologous CAR T cell therapy, given the recent approvals and the number of products that are available in the market right now. These efforts follow the success of AGene, which is the sister project of ACEL, which focuses on AAV vectors, as well as AMAB, which was instrumental in allowing advances to occur in the monoclonal antibody industry. We're very fortunate at ARM to have a large number of subject matter experts from our member companies. There are many expert contributors for this project from more than 30 ARM member companies, and this project was also supported by Nimble, who generously provided funding, which allowed us to employ technical writers for this project. The document is intended to benefit the entire field, so it is available as open access in our website at the link provided here. And this is the contents of the document. As you can see, it covers quite a big scope. From regulatory considerations, comparability, standards development, to the foundational principles of QBD, starting materials, and today's talk will cover the elements of facility design and manufacturing operations that are laid out in Chapter 11. This webinar is the 8th out of 9-part webinar series for the ACEL educational rollout. The schedules are listed here and also posted in the link down here. Our hope is that ACEL, both the document and the webinar series, can serve as an educational tool to the community, and we hope that the document will be used by product developers, educators at universities or other institutions, and we have the aspirational goal that this will be used to also help educate the regulators. And before we move on, just one housekeeping item. You can use the Q&A function in your Zoom window to ask questions, and we will try to answer as many as we can at the end of the talk. If we didn't get to your questions, we will compile them at the end and post the response in the ACEL webinar website. And with that, I'm sure you're not here to listen to me talk, so I would like to introduce our expert speaker for today's webinar. Peter Walters is a fellow of advanced therapies at CRB with over 20 years of experience specializing in pharmaceutical process and facility design. He has a deep technical background in designing processes, equipment, and spaces for multi-process facilities predicated on maximum flexibility, logistics optimization, and technologies that reduce costs while allowing pipeline expandability and higher quality therapeutics. He has gained a unique purview into aseptic equipment design and use, process scale-up, pilot and GMP manufacturing operations. His expertise includes experimental design, process characterization, equipment and process troubleshooting, and manufacturing for clinical phase 1 through commercial production. He has designed process facilities for monoclonal antibodies, gene therapies, cell therapies, aseptic novel processes, both in stainless steel and single use. He has helped author an NDA, participated in FDA facility inspections, and is a patent-awarded co-inventor of a novel process formulation method. Peter was recognized by Consulting Specifying Engineer with the 40 Under 40 Award in 2018. Peter, thank you again for taking the time out of your busy day today to be here today, and I will turn it over to you to start your presentation.
Yeah, thank you for that. Give me a quick second to get the screens set up here. Okay, hopefully that's coming through. Today, as Josephine said, we're going to give a highlight of chapter 11 of the ACell document. I think fantastic document on the whole if you haven't had a chance to take a look at it yet. And chapter 11 really tries to give a high level overview of manufacturing strategy, design considerations, kind of an over 10,000 foot look at how you go from a business case to a translated facility, some strategies around clean room design, specifically for cell therapies, and then some of the high level basics and general care and maintenance around clean rooms and all that sort of comprises that. So there's a lot of topics involved in it. We're going to touch all of them a little bit, but certainly try to leave some time at the end for Q&A, and then yeah, we'll see where we go. First off, definitely special thanks to the other co-contributors to this chapter, Jasmine and Conti. I think it was a tough discussion trying to sort of recreate how you design and build, and then the strategies involved for a facility from scratch. And so we had a lot of discussion around how to do that, and definitely appreciate not being the only person on that team. So the first section is going to give a kind of a high level, again, a very high level overview of some of the steps and considerations for just going from facility planning through the process of design into the process of what we call realization. So the construction, the building, the bringing up of a practical GMP facility. Some of this isn't specific to cell therapy because there's a lot of things about these buildings that are sort of synonymous with GMP operations in general, but try to touch on, certainly as we get into design considerations, we'll start talking very specifically about cell therapy and autologous cell therapy in general. So when we talk about facility design, certainly you aren't just picking up a piece of paper and drawing a permit set that you're going straight to the city for. There's a lot of steps and stages that get involved. Usually we see, or certainly recommend, starting with something called like a feasibility level design. It's kind of that very initial business case where you look at all the requirements for the facility that need to be accounted for and accommodated. A lot of times this is still in the business planning phase, and you're doing scope definition. So what is it supposed to do in case of like an autologous cell therapy? Like how many patients is it serving? And this starts to define how big the facility is, how much square footage you need to sort of account for in terms of the warehouse, the admin space, the QC space, the manufacturing, all the support functions, how many people are intended to be on site. So you can think about parking, amenities, all of these kinds of things. And I definitely don't want to, although it's usually the least resolved of all of the different phases of design, the pictures on the right sort of try to illustrate how you take these big blocks and you sort of resolve them as you go through the design process. The importance of that phase cannot be understated, because a lot of times in that feasibility level, you're getting your first cost estimates and square footage estimates. And the importance of getting those accurate is so important, because a lot of times that design phase is what you end up going back to your board with, or you go back to your team with, to sort of decide, is this the right case? Is this the right site for what we want to do with our business moving forwards? And I think, unfortunately, I've seen a lot of different situations where companies have dramatically underestimated the square footage requirements, or dramatically underestimated the cost requirements for a facility. And if you don't find those out until you get later in the process, not only have you invested a lot more money and time into a process that might stop short for you, but you end up with a completely different business plan in terms of how you want to bring these patients, or these therapies to bear, to bring to patients ultimately. So I think getting good partners in place early on and getting good cost estimates and good square footage estimates is super critical. And then once you kind of have that in hand, you go forward into further stages of design, concept or basic design, and then followed by detailed design. And the idea is being that sometimes you're making new decisions and you're going back and changing things. But for the most part, you're trying to take those big block decisions and shape them and resolve them into more and more detail. With more and more detail, you're getting more and more accurate estimates for how the design looks and feels and will be operated, but also cost and schedule for how long it'll take to build and how much it'll cost you in the end of the day. And of course, the idea being that changes, especially big changes made later in design, are going to cost you more, because you have to redo all of that resolution of design. So as much as you can, identify things early that you don't know. If you're still hiring your team and you don't have a good staff estimate for how many people are in the building, that's fine. But identifying that and then trying to make educated guesses that get more and more detailed as you get further in. And then certainly once you get down to like detailed design, those are going to be where you're creating documents that you're going to the city with to get permitting, and you're kind of ultimately having construction documents come out of that will then act as the building footprints for what will be your facility. I think one thing I definitely want to bring up here in the design portion of it is having foresight into your plans for how to manage QRM, QRM being quality risk management. Like how are you approaching the operation of the facility? What are going to be the practices to manage contamination and cross-contamination, and ultimately bringing a quality product to bear? You want to make sure that those aspects are all baked into the facility design as much as possible. Then you sort of have the flip side of the process, which is construction. There's a lot of different ways you can go about construction, and I'll talk about that in a minute. But ultimately, you're taking those plans and you have a site and you are building the facility, or you're renovating a space, or you're retrofitting a core and shell or something to that regard. You're going through site walks as construction's going on to make sure that construction is happening per the plans. You want to make sure that you don't end up with the facility being built and then something was designed correctly or built wrong, or that something was designed wrong and wasn't discovered until it was all fully built. You want to do field walks as much as possible to make sure that things are coming along. Once the site is mechanically complete, as we call it, you still have to start it up. You still have to make sure that everything runs properly and runs as intended. So there's a phase where you are, slowly turning on all the mechanical equipment, all the process equipment, all the utilities, and making sure they're getting into states that are operating as intended. You're doing engineering runs and engineering tests to make sure that things are to the quality levels that you want. And then ultimately you're going into your, we sort of shorthand call CQB. So commissioning, qualification, validation. That's the process of making sure that the equipment is doing what it's supposed to be doing, that it's the correct equipment per the design, and that ultimately your facility does and performs as it's intended to do. And that'll be the basis for a lot of what we think of as a GMP space and a GMP facility is basically ensuring you have control over it and that you are performing it per your validated state. When we get to CQV, a lot of companies do decide to outsource the performance of that. So they might bring in a contractor and do all the validation or the qualification studies. And that's a means of helping keep your own site staff down, certainly, but there's a number of different ways for how you might want to perform any of these steps. When we talk about construction, certainly degrees of modularity is a very popular subject right now. I think historically, what we would call stick building, which is going to be your studs and drywall sort of walls and doors. That method was the kind of standard way to deliver a facility. You have to have your site selected, it has to be prepped, and then teams come in to erect the walls on site from scratch. And it's certainly a very flexible way to do business, but it also takes a lot longer. Everything is done completely in sequential. And over the last, certainly decade, probably longer, we've seen a trend, especially in Europe, moving towards modular construction, which is where you are building, and not just clean room components, but also mechanical components, skidded systems. You're building a lot of things off-site and then bringing them on-site constructed, or at least in parts that you can assemble very rapidly and quickly. And the idea here is that you tend to compress your installation and construction schedule significantly because, again, you are building these things in an off-site factory that is intended strictly for that purpose. And then you can be doing that in parallel with your building site being prepped, your foundation being prepped, your core and shell being prepped. And by the time you go to start your clean room construction, for example, now you have a set of pre-fitted components, or in some cases, what I'm showing here is a volumetric modular clean room. It's a fully constructed clean room that's ready to just literally push into place. That of course dramatically shrinks that portion of the schedule. And so when lead time is a significant issue for your business, speed to market, moving along this sort of run of modular construction can dramatically improve that. However, you also have cost consideration. So typically, stick building is the least expensive way to bring up a facility. What we call clean room panelized systems, which is where you take the wall panels, doors, and the components of the clean room and break them into interconnectable pieces. Those tend to straddle the cost and schedule benefits categories. Both of them, they're a really great middle run solution that is very popular right now. And then going all the way to a full volumetric modular clean room, which would be where you have a fully built clean room, or a clean room that breaks into very few components, that's put on a truck and is shipped to your site and is more or less like pushed in place and then reassembled. Those are going to be typically the most expensive way, but again, the fastest. And there's also some other benefits to each of these too. One of the things about a volumetric modular clean room, for example, is you can be pre-testing the HVAC in the factory to sort of make sure that things are going to run as intended. And then you're doing confirmation runs once you get it installed on site. So a number of different ways to still compress your schedule. But yeah, different things. And I think, again, this is the kind of thing that you'd want to bring up in that feasibility level of design, certainly by basic design, because you need to design your facility to accommodate these. You can't just design for anything and then build anything. I think when it comes to flexibility, stick built is probably still the most accommodating. You can make very late stage changes in your design if you think that that's a part of how you're going to have to realize a building. You really don't know how many patients per year, or you don't know exactly the technologies you want to use for your systems. Stick building allows you to change, if you got to change a wall or you got to add a door, you just have the contractors make that change kind of in real time. And so it allows and accommodates very late stage changes very well. Whereas a panelized system, or certainly a volumetric modular system, if you're making very late stage changes, you got to go back to the factory for more components, and that kind of puts you back in line for the lead time of those. So a little bit less flexible in terms of construction. And then when we're thinking about volumetric modular, certainly some very real dimensional constraints for designing for those, because they are large pieces of equipment. So you kind of have to chunk them and block them into what will fit on a moving truck. And then you got to have access somewhere at the building exterior to bring these into the interior. You got to have room within your facility to slide them in place. And so you got to be very pre-planned in terms of how you want to use these. And then when we talk about startup and CQV, I'm showing the Gantt model here of sort of the more and more specific specifications on the left-hand side, which ultimately drive towards your construction and that initial startup steps, and then the qualification period that comes back out of it. And I think, without getting into too much detail, the point I'm just trying to bring home here is how you have very specific design requirements, the top being your site requirements or gross system requirements, really driven by operations. They get more into equipment design and more into instrumental design and very specific things. Down at the bottom, you're looking at model numbers, whereas at the top, you're looking at like how many liters you need to be able to do things with and how fast you need to be able to transfer things with. And then your qualification process is driven off of those design requirements, ultimately getting you to a place where you can then focus on process, bringing your process into the building and performing it and doing engineering runs and getting ultimately to a GMP manufacturing kickoff. So let's get into the actual clean room space and some design strategies here. I think the next couple sections are going to talk about space design strategy, and then even just like what is a clean room and how do we use them and what requirements there. I think certainly one of the things that's very tricky about cell therapy, GMP cell therapy specifically, is that there aren't any great standard models that you can use to design your space, because there's so many nuances to cell therapy in terms of what technologies you're using and what steps you're performing and how long those steps are. If you're shipping fresh or frozen, there's a lot of different considerations which resolve themselves into a whole smattering of different facility types. And I think as we're talking about autologous cell therapy specifically, as you scale up into larger and larger patient populations, any inefficiencies in your space design, any inefficiencies in your equipment usage and all of that gets scaled up with you, and you could end up with a very large bloated facility that's very inefficient and very expensive to run. And I don't think that's where anyone wants to be operating in when they're at a commercial step. So certainly as we get into like clinical 2 phase type of facilities, you want to be very mindful of how your spaces get used, because you're probably designing the footprints that will help inform your decisions for like later stage design and ultimately commercial facilities. So looking at kind of that autologous immunocell therapy type of facility, kind of the first step is to really not just understand, but document your process. And I know that sounds maybe very obvious, but there's a lot of nuance to it that has to then inform your facility design. Things like your closure plan and what kind of equipment and technologies and platforms you're planning to use to manufacture and generate these cells. That has a very real impact on the space classification, the footprint, how that manufacturing space plays in terms of when operators have to move cells from what system to what system. You want to be you want to be aware of what steps are being performed in what order and the utilization of those systems. Are you going into incubators? Are you going to wave reactors? Are you going to, using a G-Rex type system or flasks, or are you using a Milteni Prodigy? Like there's a lot of different options for how to be doing some of these cells, and they change the equipment type, they change the durations of what's in what steps, and that ultimately gets reflected in your equipment selection and your space design. Understanding for each of these steps kind of what degree of segregation is required, and if there's any high risk steps, any high risk materials, especially from a cross-contamination perspective, that needs to be accounted for, and ultimately feeds into your risk management plan. Because again, how you perform this manufacturing, you got to be very wary of chances for cross-contamination, chances for contamination from the environment, and then putting steps and operations and equipment in place to help manage all of that. Certainly, if we're talking about autologous cell therapy, you got to be very mindful of multi-patient spaces as well as multi-product. Is this space intended to just do one process for multiple patients, or is it intended to do multiple products? And what degree of segregation do you need to put in place there? There's a lot of text, especially from the Europeans, around protecting against mix-ups and cross-contamination. And I think that's kind of the core of any good, QRM policy, which ultimately wants to be reflected in not just the equipment, but in the facility. So we're going to get into a couple different strategies here. There's a number of ways that you can go about it, but these are the sort of 3 basic strategies that we talked about in the chapter. So in terms of room and space design, one strategy certainly is dedicated production space. This would be where you have a room where you are doing a process, whether the entire process or a portion of it, certainly you're doing a process in that room by itself. In this case, it would be, patient dedicated, product dedicated. This, of course, gives you maximum chance of managing cross-contamination or risk with any other steps that are going on, but they're also typically inefficient because everything in that room, including the square footage of it, gets tied up for that one patient. So these tend to be good for short duration processes. If you have a process that the whole thing can take place in a couple of days, this could be a great way to manage that process and de-risking. It's good for open processes where you can right size the super highly classified environments. It's also good for processes with very high risk steps or very high risk materials. But I think where we tend to go is that this style of design strategy can be a tool to be used in conjunction with some of the other examples I'll show in the next couple minutes. So you might not have a facility that's solely dedicated rooms, but you might have a handful of dedicated rooms, kind of as shown here, as a part of a larger facility design so that you can bring steps or processes into this kind of as necessary. This can also be great for very early clinical, where you maybe have a very low throughput facility and you want to de-risk. But again, if you're thinking about commercial spaces and larger patient populations, this sort of strategy used by itself can really sort of create very large facility structures. They're going to be very, very costly to both build and operate. Kind of on the flip side of that is the idea of what we call a ballroom. So a ballroom is going to be a much more open, flexible space where you're handling lots of different patients at the same time, maybe even different products, depending. It tends to be not only flexible because you can rearrange things in the room kind of as necessary, but it can be a very efficient space usage because you're minimizing the number of airlocks and circulation corridors per patient. Because you have all these patients in the same space, it works much better with closed processes. By a closed process, I mean a process that is fully contained within its equipment, and whereas the process stream doesn't interact with the surrounding environment at all. And so, if you've got a closed system, then you're de-risking at the equipment level. And so that can enable you to use a ballroom setting, which again is going to be a lot higher efficiency, a lot more flexible in terms of accommodating different technologies in the future, or maybe even more patients per year in the future. You can also use dedicated rooms, like I just showed in the previous slide, in conjunction with ballrooms, so that you could have, if you have one step that has to be performed open or one step that's a very high risk, you can take your cells out of a ballroom setting into a dedicated space to do that one step or a small handful of steps, and then bring it back to the ballroom. And that's a way that you can sort of manage the risk for high risk steps while also allowing for a much better sort of efficient, flexible space. If you're going to use a ballroom, certainly for any number of steps, one thing you're going to want to plan for is how you're managing that segregation, if it's at the equipment level, for example, and separation. Because again, it comes back down to preventing mix-ups and cross-contamination between patients and products. We also introduce a strategy in the chapter that we sort of called a dance floor. And the idea here is using a combination of different rooms that are sort of right-sized to unit operations, and as opposed to like performing something in one space, maybe bringing it to another for certain things and then coming back and having like workstation types approaches. The idea of a dance floor is to basically have spaces that are dedicated to certain unit operations, and you're moving your process across rooms through those unit operations. I think what's powerful about this approach is that you can right size each of those rooms and number of equipments to sort of accommodate utilization needs for that step. So if you spend a lot of time in an incubator, for example, you can have a space that's a little bit less classified with a lot of incubators. And if you're spending a lot less time in like a separation system, for example, you could have fewer separation systems in the right classification to support that. And so this allows you to kind of really drive up utilization in your equipment, which ultimately is a much more efficient space design. However, in this type of strategy, you are moving your process through the building somewhat significantly. And so you're going to have a lot, maybe more movement logistics in the hands of operators to move those things around. You're going to want to make sure you've got practices and procedures in place to not only navigate the transitions maybe between clean rooms, but also in terms of chain of custody and chain of identity, that you're not mixing up different cells as you move through that. Ultimately, with whatever facility design you're going to go with, you're going to want to map all of your GMP operations. I've got a couple different colors on the screen here to sort of show some examples of how you might map personnel by gowning level. You might map how your process moves all the way to your drug product. You might map how you move materials or samples or certainly waste. Looking at these so you can help identify traffic patterns through your facility and making sure that you don't end up with bottlenecks at things like airlocks, but also make sure that if you have a lot of movement through certain corridor areas, you're aware of that and you can put things in place like temporal segregation, or maybe you make the corridor a little bit wider, or you've got staging spaces to help accommodate those movements. Because again, if you have a lot of motion, that introduces the opportunity for mix-ups or contaminations. And I'm showing sort of the GMP cleanroom cores here, but you should look at things like material and personnel movements across your whole sort of life cycle of that process. So where it comes in at shipping, receiving, through the warehouse, through the facility corridors, into your clean rooms, and then back out to make sure that things are moving, especially for autologous cell therapies, things are moving through your facility in a very facilitated way, so that you don't end up with mixing up of incoming and outgoing materials, for example. When we're talking about clinical one spaces, I think the idea of using a glorified lab works pretty well, but I hate when people refer to GMP facilities as like just a lab, because there's, again, the building and the space design wants to facilitate all the movement. So I have a quick baseline case study here to sort of show two examples of how using the same square footage, you can dramatically change how the operators interact with the process in the space. So in this example, you've got what is sort of thought of as a unidirectional flow suite. We have a ballroom with various biosafety cabinets. You've got shared equipment, which could be in-process testing, cell counters. You've got maybe incubators or freezers or refrigerators kind of put at the two ends. And so at a room level, you've got people coming in one side, going out the other. But within the room, you sort of get this spaghetti diagram of back and forth movement between the workstations and the shared equipment, and people are overlapping, and it's definitely not unidirectional flow within the space. Definitely some opportunities for people sort of across mixing up materials. Whereas if you take all that shared equipment and you sort of lay it out longwise throughout the center, and you shrink the footprint top to bottom, so now actually the square footage is identical between this last model and this model. However, you can see not only do you have unidirectional flow through the room in terms of people coming in one side and leaving the other, but you've now separated that central aisle into two, and now you get people bouncing back and forth from their workstation to that center place and out. So it's a much more facilitated unidirectional flow, much better segregation within the suite, much more identifiable workstations and process flow. It's kind of like an artistry to how you do these. Ultimately, you want to get to a general arrangement diagram that's very specific in terms of how you lay out your equipment and your room flow. Certainly, you want to be mindful of how many people are going to be working in this space, what utilities are needed, and trying to make these, especially when we talk about autologous cell therapy, trying to make these as efficient square footage wise as possible, because usually the idea around autologous cell therapy design is you design for X number of patients through kind of this like workstation approach, and then you scale these out. And so any inefficiency in utilization of equipment, additional redundancies that maybe aren't needed, certainly any dead square footage in this space is going to duplicate every single time you repeat this out. And so getting it right the first time is definitely critical as early as you can. At the same time, you kind of want to be mindful of designing for a way to manage change, understanding that a lot of these processes, especially in early clinicals, aren't quite nailed down in terms of the technologies they're planning to use. And so being mindful of giving at least a little bit of space to accommodate different platforms that might come in, or different technology changes as you go through that maturity life cycle of, clinical and realization into a commercial setting. And again, I think ultimately you want to have a facility design that is trying to prevent any chances of mix-ups and cross-contamination. There's a couple different design guides out there. We tried to capture a couple things in the chapter 11 itself, but as an example, NIH has a great guide for biosafety cabinet placement, some kind of rules to help follow to make sure that you're not getting crosstalk between various biosafety cabinets, that you're not putting things so close together that you're impinging on their processes. And so we always try to incorporate these as we lay out, again, the equipment, the support equipment, the testing equipment, but also the primary manufacturing equipment with any biosafety cabinets that they need to be interacting with. Another kind of facility consideration, the EMA released the latest update of Annex 1, which I think certainly as far as the facility goes, has some implications for how you would design cell therapy facilities. And there is a strong push now towards the ability to permit observation into production areas where you're doing aseptic operations. So really thinking about that glazing in the glass, not just for opening up daylighting and showcasing for the operators or showing off your processes, but also from a like regulatory standpoint, they really want the ability to see into the space and get eyes on it without having to do, you know, full bunny suit gowning and getting into it, which is not really where you want people in terms of like observers or anyone from the agency to really be all the way in there anyway. And then kind of the last thing about sites, when we talk about the facility on the whole, how you arrange all the spaces, especially for autologous cell therapy, is pretty critical. You want to make sure there's good handoffs between your shipping and receiving and how that gets to the warehouse, or how your apheresis gets to its inspection area, how that gets to the manufacturing, how the QC samples get handed off. And so how you arrange all the components of the whole space should be methodically thought through, really thinking about chain of custody and chain of identity throughout the course of it. We'll get into classified spaces. I'll try to move through some of these pretty quick. I know some of it gets kind of dry, but I think there's some things that are worth touching on. And again, we can always get back to, or you can always refer to the chapter if there's any questions that we don't have a chance to get to. But originally for the US at least, the use of federal standard 209E was used. This SKU here, when people say class 100 or class 1000 or class 10,000, I think the only reason we mention it now is if you are using those terms for this standard, this was replaced, you know, over 20 years ago at this point. So it's definitely vestigial, and you want to be moving towards the much more modern standards. The International Organization for Standardization released ISO 14644-1, which isn't specific to biotech, but is specific to clean room standardization. And you can see there's this categorization of 1 through 9, 1 being the highest degree of clean airflow, 9 being sort of the least clean of a classified space. The US FDA points directly to the use of this standard for how you define your clean room settings. And so we typically see like ISO 5 through ISO 7, ISO 8 in reference with biotech facilities. Now, for the EMA, UDRILEX points to specifically the use of the ISO standard as well, but incorporating the numbers from that standard into a little bit more biotech focused overlapping standard that they call the grade system. So grade A, B, C, D, A being the most stringent kind of clean room or clean air setting, D being the least stringent of a classified space. I've tried to show in the color bands here how they interact with each other. I think one thing that is worth noting is that the ISO system doesn't account for whether there are people in the room or not in the room, what is happening in the room. It is just purely a standard for air quality, whereas the grade system really tries to look at the degree of air quality while operations are going on versus at rest, which is when there's no operations going on. And you have to accommodate both those situations. And so you can see how there's a step change, excuse me, there's a step change in that particle count between in operation and at rest. But typically speaking, how we would translate between these would be around the in operation standpoint. So grade A pretty much relates to ISO 5, which would be the airflow around critical operations that we have open cells that you're manipulating. Grade B or ISO 7 would be the background to that setting. So if you have a biosafety cabinet that's got a grade A environment, you'd have a grade B background to that. And then grade C or ISO 8 is sort of your general clean room core for any kind of at-risk activities. And then grade D is going to be much more de-risked. You'll note that there isn't an official grade D equivalent in the ISO setting. So sometimes we refer to that as CNC for people that are doing purely US type facilities. Although for the Europeans, you can't have a CNC below grade D even as well. Really quick to kind of go through some case studies. So again, if you're doing like an open manipulation of cells in a biosafety cabinet, that biosafety cabinet's going to need to be a grade A, and then the background to that is going to need to be a grade B. And that's pretty much aligned with the US. They point to an ISO 5, which is kind of the equivalent. I've got that room classification table there in the bottom right to help translate between the European grade system and just the ISO. So an ISO 5 biosafety cabinet with an ISO 7 background. Now we're talking about isolators. It gets a little bit trickier in general. So in Europe, I think there's now defined open and closed isolators, which changes the backgrounds you can be using. I think in general, Europeans are a little bit more open to the idea of going to, if you have a fully closed isolator, you could go down to a grade D background. However, because there's no ISO equivalent for a grade D, the US and the FDA is still a little bit hesitant to how you manage that. They still want to see that the aseptic operations are being performed in a clean room setting, and that typically comes down to an ISO 8 background. However, we have seen in some type C meetings the discussion of driving towards, if you're fully adopting the grade setting, that you can maybe get down to a grade D with the US regulatory. So it still is a little bit of a nebulous area, but I think that's trying to get navigated. And then again, with the Europeans, a difference between an open isolator, which would be an isolator with a mouse hole or a transition area, versus a closed isolator where everything is fully contained. I think if it's fully contained, you can get down to a grade D background. If there are openings in it, they still want to see a grade C background, which makes sense from a risk standpoint. And then for any sort of closed systems, I think you need to be assessing how robust the materials are in terms of if you if you want to push to a grade C or a grade D. I think certainly in the US, again, they're going to want to see all that in an ISO 8 background. So we tend to just design around a grade C. I'm hesitant to bring this up, but I think it's just more around there are some exceptional circumstances for which in very early clinical, if the process just cannot be, by the means and definitions of the process, it cannot be done in a fully sort of GMP environment, that there are some considerations you can do. I think certainly you gotta still perform it in something like a biosafety cabinet, but you could have lower classified backgrounds. Again, if something like it had to be performed immediately next to the patient, or some sort of circumstances. I think we talk about autologous manufacturing for the most part. I have never come across this to date, so I'd be very hesitant to point to this as an exceptional case that you could do. But I think in general, the idea being mindful of your process and the steps involved in it, and then trying to accommodate the regulatory system as much as possible. A real quick look, I know we're getting close on time here, a real quick look at kind of some final considerations around facility use and planning. So we talk about clean rooms. Humans are the #1 particle generator by far. And so being mindful of, as you go into higher classified areas, not only are they more expensive to build and maintain, but you have to be doing higher degrees of gowning. And all that gowning is there really to protect the clean room from the people, try to contain as much of that as possible. You also have to qualify and monitor these environments to make sure. So having a site plan that kind of walks through where you're sampling and how you're sampling and how often you're sampling, to ensure that your clean room backgrounds are being maintained at the levels that you're claiming. Higher classified areas are going to require more and more frequent samples. So like an ISO 7 or a grade B space, you're going to want to sample more often than ISO 8 or a grade D space, for example, to really show, because it's going to be a higher critical area. And then certainly what we call the critical zone. If you're doing any open manipulations in an ISO 5 or grade A space, you're going to have to be monitoring continuously throughout the entire course of that to make sure that it was a qualified environment for the entirety of that manufacturing step. And then just being mindful of, if you want to comply with UDRIL-X and the grade system, you got to do some form of sampling for both in operation and active usage, but also passive sampling for at-rest considerations. And then again, all of what we just talked about comes back to that QRM principle of quality risk management and making sure that your processes, your procedures, your equipment, your facility is all sort of designed and put in place to manage the underlying risk for your process, and making sure that you're kind of doing your best to bring forth the highest quality products for patients that you can. So that's what I got. Certainly, thank you for your time. I think we probably have a little bit of time for Q&A, Josephine, but if there's a question that we didn't get a chance to, you can certainly reach out to me directly and I'll do my best.
Great, thanks so much, Peter. Yeah, I think we have a couple questions coming in. So I'm gonna go ahead and jump to the Q&As. Actually, I just want to remind everyone first, I saw a couple raised hands on the chat. So if anyone can use the Q&A function to ask their questions, that'd be great. So first question, let's see. So we're seeing facilities that are accommodating different modalities now, different cell types, different vectors. And, you mentioned earlier that they have different manufacturing and different safety levels, and you mentioned segregation within the facility. But I think one thing that wasn't discussed too much is the different timelines possibly, and how fast these different products move through the different pipelines. So how do you incorporate this into your design consideration?
That's a great question. I think we refer to that typically as like a multimodal space, which is a space, whether a whole building or sub facilities or whatnot, that have to accommodate, whole different platforms of manufacturing. And I think cell therapy and a gene therapy or a viral vector is a great example there. I think where we are seeing most of the industry going at this time, again, understanding that industry is kind of going through its maturity cycle of growing and exploring, but also the back and forth with the regulatory agencies. I think most companies are seeing success with having, you can have one building that does both like viral vector manufacturing and cell therapy manufacturing. But I think we tend to see two separate sub facilities within that building. So separate gowning, separate air locking, separate corridor systems. One kind of bespoke to your cell therapy and one kind of bespoke to your gene therapy or your viral vector components. And that allows you to sort of right size those in a way that also de-risks them from each other, and definitely limits the chances of cross-contamination if they're in fully sort of segregated areas. I think that is not to say that you can't combine them on a corridor system, or that you can't push them really close together, that you couldn't even have suites that flip-flop between doing one or the other. But I think the closer that you push them, the higher risk there is of cross-contamination or mix-ups. And so the agency is going to want to dig into those a little extra closely to make sure that you have additional things in place to show you've got turnover, and that you've got cleaning that's appropriate to, your flows are appropriate to really delimiting that chance of cross-contamination. I think, again, through the course of industry maturity, most companies just don't want to have to manage that risk conversation with the agencies right now, and so they do separate facilities. But again, in terms of utilization, it's probably not often that, like, a dedicated suite for doing autologous cell therapy, for example, is going to be hyper useful for doing any sort of larger scale vector manufacturing. So if you're doing very small scale of everything on benchtop scale, then I think there's a lot higher chance that you could cross utilize. But if you're talking standing bioreactors versus autologous cell therapy, I think from a utility standpoint, from a utilization standpoint, it just makes more sense to kind of design those spaces bespoke to each other anyway.
Great, thank you for that. Can you comment on the potential application of these principles to decentralized manufacturing, the potential use of closed system for production at a clinical site?
Yeah, great question. Yeah, certainly decentralized manufacturing has been a growing buzz within the industry. I think the the principles behind these GMP requirements, especially like classifications and whatnot, those are going to have to stay in place. It's just the scale of them is going to change, because now instead of having one facility that does 5,000 patients per year, for example, you're going to have however many facilities that does a portion of that. I think where we've seen success already at a decentralized model is partnering with academic hospitals or research hospitals where they have an in-house, they're putting in an in-house clean room environment. So it's going to be a much smaller area where you are still gowning up, you're still going into the appropriate classifications so that the risk around that operation is still managed appropriately. Now the question mark is, how do you manage all the logistics that surrounds that in terms of quality testing and supply chain and centralizing your QC release, your QA release, like all of that. There's still a lot of questions around there. I think definitely if you're pushing towards or thinking about decentralized manufacturing, moving to manufacturing equipment platforms that are more closed and automated really helps de-risk the chances of operators being a course of differences between those sites. And the more closed you get, the smaller you can make that clean room setting and the less risk you're putting on the facility. And that starts to get into much more flexible, decentralized spaces for sure.
Great, thank you. Let's see. Parallel processing for autologous therapy is key to scaling, and assuming the manufacturing processes are closed, what are your thoughts on parallel processing of different patients in different therapies in the same suite, assuming appropriate analytics are in place to differentiate the products?
Yeah, I think if your equipment systems are fully closed, I think the regulators are a lot more open to doing more and more patients in the same space. And then it becomes a question of how many patients within the same ballroom setting are you comfortable with as a business? Because obviously the more you put in there, if there's an issue with the clean room or an issue with one of the batches where it leaks out or something like that, you've now potentially implicated all of those batches in that space. So I don't think companies necessarily want to go to one gigantic ballroom where they're processing 1,000 patients, because I think that's still a lot of risk to manage with one room in general. But definitely, I think there's a lot of de-risking at having a fully closed process platform and doing lots of patients in one space in a very efficient, flexible manner. I think in terms of doing different products within that space, again, you're going to look at what are the materials that are different between those products and can they be managed in a closed way? Because as long as you keep the clean room environment sort of agnostic from your process, then it shouldn't matter what components are being used because they're not interacting with the environment. And so then they're not interacting with the other patients, the other systems. But you will want to be managing things like in-process testing and making sure that even if you're taking a sample in a closed way, how that sample gets prepped and primed and tested isn't open in the room. So maybe you have to put your cell counters in biosafety cabinets or things like that, because that can implicate the airspace, for example, at least from a regulatory standpoint.
Mm-hmm. So if we can go back to the segregation discussion earlier, how would you segregate areas working on virus from non-viral areas within the same facility?
Yeah, typically, and it comes, again, you got to think about the closure of the equipment and the chances of exposure to the environment or the operators. I think in general, a not an easy button, but a way to help manage it very cleanly is to have unidirectional flow through, especially the vector areas. So if there is a chance at a leak or anything that's getting onto operators, they're degowning in a separate space from where they gowned in, and then they're moving to a corridor system that feeds all outgoing. And that separates that process almost entirely from anything incoming that might implicate adjacent spaces that could be cell therapy or maybe a different viral vector. That tends to be a way to really manage the GMP flows of, again, materials, equipment, personnel in a way that de-risks any of the other kind of parallel processes that might be going on in the same facility.
Great. I'm going to sneak in one more question. I know we're at time right now, but can you provide a stainless steel grade specification for carts, benches, and tables?
Yeah, I'd say you want to probably look at your cleaning regime for those. So as you get into things like sporicides, if you're doing VHP, some of the really more aggressive systems, you're going to want a higher grade of steel. I think it's kind of the same rule we use for tanks. Like 316L works pretty well for most of the cleaning things. But certainly if you are leaving high chloride materials on that are going to be, if you're seeing rusting and things like that, I mean, I wouldn't necessarily from a cost perspective think about really high alloy carts. I think you'd probably be best moving to plastics if you have a cleaning system that gets really, really aggressive in the chloride standpoint. So like Teflon coated or things like that.
Okay, so I know we're at time right now, so we're going to wrap up the webinar right now. There are a few other questions on chat that we didn't get to, so we'll make sure to get to those offline, and we'll post the answers on our website. Thank you again, Peter, for your time and lending your expertise to us. And yeah, well, thank you again, everyone. And the recording of the webinar will be posted by the end of the week. So thank you, everyone. Have a great rest of the day.
Thank you.